Hello, biology students, and welcome to Genetics with Miss Shimel. Today we're going to take a look at a Punnett square, which is a tool to be used to predict the probability of an offspring's genotype and phenotype. If you're a lifelong Bellwoodian, I'm sure you have this back in 7th grade, maybe with Mr. Hescox in life science. But we're going to revisit the Punnett square today. It's a predictive tool used in genetics to help you see the possible outcomes of a genetics cross. For example, if you have a green pea parent and a yellow pea parent, what are their offspring peas going to look like? In this case, we can tell that Y is capitalized, so yellow is dominant. Little y is lowercase letter, so it must be recessive. This genotype is going to be heterozygous, which is hybrid for the trait. But the dominant over masks, beats up that little tiny y. So this one appears yellow in color. So does this one. This genotype down here is homozygous because they're both the same, lowercase, lowercase, and their phenotype is green because it's the recessive trait. So this box over here, this foursquare, is just considered to be a Punnett square, named from Reginald Punnett. We're talking about classical Mendelian genetics, where something is dominant over something that is hidden or masked or recessive. Monohybrid just means one trait at a time that we're going to examine or look at. Probability is the likelihood, the chance that something will occur. Genotype, your type of genes. Your phenotype is your type of genes that you inherit. Which letters, which alleles did you get on your DNA? Your genotype is the type of genes. Your phenotype is your type of physical appearance. How do you look based on the genes that you did inherit? Homozygous means the same. For example, if you're capital A, or if you're little a, like in this case, little y. Heterozygous is capital lowercase, like in the case of these yellow offspring, or even this yellow parent right here. See that yellow pea pod? Your alleles are the letters that we use to represent the DNA, the genetic code that you have received, and capital letters represent the big, strong ones. Recessives are lowercase letters. Dominant will hide or mask the recessive traits, like in this genotype that is hybrid or heterozygous. I hope from your classes here at Bellwood that you know that probability can be expressed in various ways. For example, you can use fractions. For example, if you flip a coin and you get heads versus tails, you might get one head for every tail. So it's going to be a 1-to-1 ratio, or 100% of the time, or it could be 1 out of 2.5, 50% of the time, and you would say 50-50 or half. So you can use ratios, you can use decimal points, you can use fractions, you can use words to express that probability, that prospect, the likelihood, the chance of something actually occurring. Here's an example of what it means to be homozygous if we're talking about classical Mendelian genetics. If you get two of the same letters, whether they're capital or lowercase, for example, this is homozygous, as is this. But in this case, if you're talking about dominance, you get the two capital letters. This one, on the other hand, is still homozygous, but it's homozygous recessive. So here are different examples of, is this heterozygous? Is this homozygous? Is it hybrid? Is it mixed? Or is it purebred? If you are homozygous dominant for a trait, what will you pass on to your next generation? Let's make you purebred for being from Bellwood. If you go through interphase before meiosis or mitosis, that would double your content, double your alleles, double your genetic information. If we do meiosis 1, then meiosis 2, you would only pass on 1/2 of your genetic information. If you started with homozygous dominant, you could only pass on the capital letter B in your eggs or your sperm if you did oogenesis or spermatogenesis. So what we want to do is use this little tool over here called the Punnett square to show you what are the likelihood, what are the chances of you passing on this capital B or this capital B. You just bring the letters down. Bring the letters down on the Punnett square. So if this is your genetic makeup, if those are your two alleles, you're homozygous dominant for capital B, you're from Bellwood, mom said to be from Bellwood, dad said to be from Bellwood. The only thing you pass down to your offspring, 1, 2, 3, and 4, is going to be that capital letter B. If you are recessive for a trait, what will you pass to the next generation? This time we're going to make you homozygous recessive. If you do interphase, you double that, so now you have 4 alleles. If you go through meiosis, you can only pass on half of your DNA to the offspring. What are the chances you would pass on this B or this B or this B or this B? A good way to do it would be to show it here on the Punnett square. So if this parent is little b, what's the only thing they're going to pass down to their 4 offspring? Just the little b. That's how you fill in the Punnett square for one parent who is homozygous recessive. This time we have a homozygous dominant parent from Bellwood, and they're going to get together with someone maybe who is homozygous for Tyrone. So if Bellwood and Tyrone get together, they would have all 4 offspring getting both the Bellwood gene and the Tyrone gene. Again, you can see that the capital B just carries downward, and the lowercase b's from the recessive parent carry across, and you would see that all 4 out of 4, 1 out of 1, which would be a 100% chance of being dominant, also being capital B, lowercase b, being hybrid for the offspring. All of these will appear as if they are from Bellwood, but their alleles say that they're Bellwood-Tyrone mixed. So they are dominant because we're talking about classical Mendelian genetics. They are all hybrid. The genotypic ratio in here is going to be 4 to 0 or 1 to 0 because everybody's the same in this case. So this Punnett square is just another example to show you if you have one homozygous dominant parent gets together with a recessive parent, all of the offspring, all 4 will end up being dominant but hybrid for the trait. They get the capital letter for the one strong dominant allele and the lowercase letter for the recessive allele. Their phenotype is still dominant, whatever that is. In this case, A for awesome instead of lousy, little a. Their phenotype is awesome, dominant, but they are hybrid for that trait. When both parents are heterozygous, we're going to see capital B from Bellwood, but with the Tyrone gene, capital B from Bellwood with that Tyrone gene. What are the chances that their children will be awesome Bellwoodians like we are? Right there's 1, right there's 2, even though it's hybrid. Here's a third, still a Bellwoodian, but this 1 out of 4 will be from Tyrone. So you get a new ratio we didn't see from before. You get 1 to 2 to 1 for the genotypic ratio. There is 1 that's capital B, 2 that are capital lowercase hybrids, and 1 that is recessive. So you have 3 here that are dominant, 1 that is going to be recessive. What new ratio should you memorize right here? Anytime you take a hybrid plus a hybrid, you're always going to get the 1 to 2 to 1 ratio of dominant to recessive. Homozygous dominant, heterozygous dominant, recessive. It's a 1 to 2 to 1 genotypic ratio, a 3 to 1 phenotypic dominant to recessive ratio. Anytime you cross 2 heterozygous parents, you should always end up with 1 to 2 to 1 for homozygous to heterozygous to homozygous recessive. 3 of these are going to be dominant. This 1 is going to be recessive. What new probability should you know? The 1 to 2 to 1 genotypic ratio and the 3 to 1 phenotypic ratio. Is it possible to have 7 children in a row who are homozygous dominant? Could you have 1, 2, 3, 4, 5, 6, 7 in a row who are dominant? Absolutely. What about recessive? Could you have 7 children who ended up being from Tyrone every time? Yes. They would not be awesome in this case. Is that possible? Yes. Is it probable? No. Remember that only 1 out of 4, a 25% chance, will be your recessive children, like this. The goal of a Punnett square is to show you the possible and probable outcomes. It's typically an empty foursquare for a monohybrid cross. You put one parent up on the top and one parent over here on the left-hand side, and just fill in the gametes to make the zygote, to make a new offspring each time. You would get 4 offspring from any possible combination. Here are 4 different examples. One example that you don't see is what happens if you cross a homozygous recessive with a homozygous recessive. If you do a Punnett square, if both parents are homozygous for recessive, all of the offspring could only end up being recessive. Thanks for watching this video with Ms. Shimel for genetics and how to do Punnett squares.